Welcome to this web brief discussing an ongoing unmet need for antibody specificity. My name is Carl Kovacs, Head of Customer Service at OriGene. In the next 10 minutes, I will present case studies on OriGene's breakthrough technology, the High-density Protein Microarray, and how it can be used to test an antibody's specificity, which is ultimately important in the development of tomorrow's companion diagnostic tests. I hope that you will be convinced after this talk that not all monoclonal antibodies are monospecific, and that UltraMABs are the future standard to which all others must compare, establishing a new best practice for the clinical use of antibodies. For most antibody applications, good antibodies are defined as those having both high sensitivity and specificity. If an antibody is sensitive enough, then you can see the results or even measure the affinity or kinetics of binding to its antigen. But what about specificity? How can we really know if the antibody has affinities to a single or multiple antigens? Well, if all protein antigens were available, and they're not, then you could test each one independently. If you add in the complexity of post-translational modifications, then you can see that the test for absolute specificity is almost impossible today. Unfortunately, there has been no technology made available to systematically address this question. So, why is antibody specificity so important? Take, for example, the anatomic pathology application of staining tissue samples for biomarkers or diagnostic targets. The resolution of a stained image is not at the molecular level, and so we rely on the antibody's ability to reveal to us the presence of an antigen within an organelle or in some membrane structure. This is a very complicated environment. If the antibody cross-reacts with an off-target antigen, then it could easily generate false positive results, which may be critical in the decision made for subsequent patient treatment protocols. It has been published that some diagnostic antibodies currently in use are known not to be completely specific, and this is primarily because there's been no validation technology to test for this. That does not mean that they are not highly valuable in their ability for use in the clinic, but if one were to choose the best antibody for such a purpose, one characteristic has to be specificity. OriGene's solution to this employs a high-density protein microarray technology that we refer to as our 10K chip array, based upon the unique number of proteins spotted in replicates. As the number of antigen targets increases, the naming of each successive version will also increase. A couple of years ago, we decided to try and address this question of specificity by taking advantage of the breadth of human cDNA expression clones in our TrueORF collection. Starting with our core competency, we systematically transfected each cDNA into HEC293T cell cultures and harvested the protein lysates containing the overexpressed proteins, tagged with both DDK, also known as FLAG, and the MYC epitope. We then optimized our spotting technology to print these overexpression lysates onto high-density protein microarrays. Of course, both positive and negative controls are necessary, and these are also spotted on every specially made slide with serial dilutions. This is a picture of one slide stained for total protein. These chips are used to evaluate antibody-antigen interactions in a high-throughput manner. A primary antibody or a serum will be exposed to this microarray chip containing over 10,000 unique proteins to see if it will interact with both the intended target as well as any off-target proteins. The second version of this 10K chip is nearing completion with almost double the content. While the interactions are usually quite obvious to see by eye, there are statistical analysis options that we can apply with definable thresholds to be certain of the conclusions. This presentation discusses two case studies surrounding the specificity of two commonly used monoclonal antibodies in the clinical setting. The first is the most popular HER2 diagnostic antibody clone on the market, 4B5. This antibody against the HER2 protein is used to type breast tumors to assess the eligibility of a patient for the Herceptin treatment, which targets that same antigen. Using the protein microarray technology, we tested the 4B5 HER2 antibody for its specificity against 10,000 unique protein targets. Remember that these protein targets were expressed in human cells and obtained through a gentle lysis procedure, and so they should be as close to the actual tissue antigen as is practically possible, especially with respect to proper folding and any required post-translational modifications. The data shown here on the protein microarray confirms that the 4B5 antibody recognizes HER2, but there is also a significant cross-reactivity with ERBB4 and ZSCAN18. The decoding of this antibody binding could not have been determined without the use of a high-density microarray. This antibody is not specific, and its cross-reactivity could result in the misdiagnosis and inappropriate treatment plan. To confirm this finding, we used the same antibody in a different assay, that being Western blot analysis. Compared to controls, we were indeed able to show the cross-reactivity with these two other proteins, namely ERBB4 and ZSCAN18, that were identified using the 10K chip. Using this same screening procedure, OriGene screened its own library of monoclonal antibodies targeting HER2, using the recombinant full-length protein purified from the overexpression in HEC293 T cells. You can see in this slide that in addition to the positive controls, the HER2 spots were successfully stained to the exclusion of any significant binding of other antigens on the slide. We were very excited about this finding and have very high hopes of employing this technology in the creation of many more ultra-specific monoclonal antibodies. Hence, the new brand name for the ultra-specific antibodies was created as UltraMABs. Of course, we can't forget about the sensitivity aspect of a good antibody, and therefore we needed to make sure that this HER2 antibody had the ability to successfully provide positive immunohistochemistry data. These pictures show it does indeed stain HER2-positive cells in breast cancer tissue slides. In fact, the upper panel of pictures was done with OriGene's UAB36 clone, and at least in these experiments was shown to be as sensitive, if not more so, than the commercially used 4B5 clone with pictures in the lower panels. We have to include that any improvement in specificity or sensitivity is good, but an improvement in both really highlights a great performance from an antibody. So what is the impact of the 4B5 cross-reactivity with HER4? We procured tissues that were uniquely positive or negative for HER2 and HER4. The tissues were typed by the original donor medical institution and then verified independently by a second board-certified pathologist with respect to the strength of HER2 or HER4 expression levels. This time, the upper panel shows the commercially used 4B5 antibody staining for HER2-positive tissues. But, as you would expect from our chip data, it also stains the HER4-positive tissue, top middle, as well as the normal stomach, top right. OriGene's UMAB36 also stains for HER2, but does not show the same cross-reactivity in either of the other slides. Therefore, in the HER2-weak and normal stomach tissues, the 4B5 antibody will incorrectly type the tissue and potentially affect the downstream therapeutic intervention. In case number 2, ERCC1 is the intended target, which is used diagnostically to determine which patients should or should not benefit from one of the platinum treatments. While the commercially employed antibody 8F1 was confirmed here to recognize ERCC1 and the variant of the same, it is also shown here to cross-react with PCYT1A, a different protein. Again, the importance of specificity in the use of diagnostic antibodies has to be taken into consideration. You can imagine how excited we were again when we screened our anti-ERCC1 library of clones to validate yet another ultra-specific antibody using the 10K chip protein microarray. You can see that this clone, 4F9, recognized only ERCC1 targets on the array, but this time to the exclusion of all others. These data, as well as other test data using head and neck squamous cell carcinoma samples, have been recently published and presented at the USCAP 2013 meeting. The pathologists concluded that the crisp nuclear staining of the 4F9 antibody showed a higher level of clarity, allowing an easier positive scoring. Again, knowing and trusting that a critical antibody is specific in addition to being sensitive makes this a preferred choice. HER2 and ERCC1 are just two examples of OriGene's growing UltraMAB collection. OriGene produces hundreds of monoclonal antibodies every month. All of these antibodies began their journey originally qualified at OriGene as TrueMABs, which were first screened for functional applications including Western blot, immunofluorescence staining, immunohistochemistry staining, and flow cytometry. Selected antibodies that passed the 10K chip test were moved forward to the next step, a clinical evaluation with patient samples. Only the antibodies that passed all of these screening steps will become an UltraMAB. Without the value created by the original source of quality antibodies, the TrueMAB collection, OriGene would not be able to provide UltraMABs, the gems among precious stones. In addition to UltraMABs against HER2 and ERCC1, OriGene has many more in the pipeline and already more than 100 available right now. Many of these antibodies are directed against cancer biomarkers and important diagnostic targets, such as beta-catenin, P53, P63, CD2, keratin 8, and SOX5. If you would like to learn more about UltraMAB, please contact us. Thank you for listening. And remember, not all monoclonal antibodies are monospecific. Why risk your patient drug or research on an untested reagent? Trust only UltraMABs, the future standard in antibody specificity. Thank you for your attention and best wishes for all success in your research.